So last, but certainly not least, we welcome Professor David Thomas with the Garvan Institute for Medical Research in Australia. Professor Thomas is a driving force in Australia and incredible advocate to the EHE medical community. He could not be with us today, but he generously pre-recorded his presentation. And so if we can go ahead and start that presentation, that'd be great.
So thank you to the organizers for the opportunity to talk to you today about our precision oncology program and how it might be useful to research into epithelioid hemangioendothelioma. About 5 years ago, we established an incorporated entity to enable a national ecosystem to bring together all the key stakeholders required for precision medicine: academia, the clinics, government, and industry. The company that we established is a non-profit. It involves 11 academic centers of excellence and 3 medical research institutes covering all states in Australia, and there are 8 states and territories. That enables us to scale up to a population of more than 80,000 cancer patients, to more than 25,000 new cancer diagnoses per year, and importantly, more than 20,000 patients with rare, less common early onset or poor outcome cancers, which are the focus of our work. In general, OMICO, or the Australian Genomic Cancer Medicine Centre, is based around the following architecture. We have 2 broad areas of the program. One is about molecular screening and therapeutics, and the other is around understanding why cancers happen and the genetics behind that. In the molecular screening and therapeutics program, we offer patients with advanced and curable cancers tumor profiling, and then link that to a basket trial of what we call clinical sub-studies. In the germline, we focus upon 2 populations, one of which I'll speak about a little bit later in this talk. The genetic cancer risk in the young study, which is focused on early onset cancers, that is, cancers arising under the age of 40, and the International Sarcoma Kindred study, which has a particular focus on sarcomas. And this now reached some 3,000 patients worldwide. For the germline risk component of this program, we undertake whole genome sequencing of a blood sample to take an unbiased approach to understanding the question, why did this happen to this individual, and in particular, why did it happen early? A fundamental principle of the program is that we use this genomic information to guide management, and in the germline risk, we have a surveillance program for patients identified as having high risk called SMOC+, or surveillance in multi-organ cancer syndromes, using the technology of whole body magnetic resonance imaging to detect cancers at an early and curable stage. And I won't talk any more about that for reasons of time. The majority of the focus in this talk will be about the MOST program. So the general principle behind precision oncology is that a molecular understanding of cancer, for example, in the case of breast cancer, breaks it up into important subgroups based around precise molecular characteristics. And those molecular characteristics, importantly, are targeted by various types of treatment. But the paradox of precision medicine is that it can take a whole range of rare cancers, and about 50% of all cancer deaths are due to rare and less common cancers, making them a particularly underserved, important group that precision medicine can impact. But we can find common molecular targets shared by different of these specifically rare subtypes, identify a common drug target, and then treat them in aggregate, overcoming some of the statistical challenges in doing research in rare populations. Of course, epithelioid hemangioendothelioma is a prototype for a rare cancer population which has suffered from the difficulties of studying rare diseases. The concept behind precision medicine doesn't need restating, so I'm going to do that very briefly here. This summarizes a meta-analysis undertaken by Roselle Kurzrock and colleagues, showing that even in phase 1, the use of a biomarker-driven targeted therapy in a phase 1 study has about 6 times the response rate, objective response rate, previously seen in phase 1 using, for example, cytotoxics or non-targeted therapies. That means the game has fundamentally changed, and it means that biomarker-driven therapies, even for new drugs where we don't know what their range of activities is likely to offer a much greater probability of benefit than other circumstances. For diseases like epithelioid hemangioendothelioma, where we don't need to begin, this fundamental principle, combined with the application across many different cancer types of this approach, enables us for the first time to start systematically enrolling patients with disease onto trials to see whether they, for either rational or perhaps unknown reasons, may benefit from targeted therapies. I'll just make one parenthetic comment here. The response rate in phase 1 to targeted therapies, about 30%, as seen in this graph, is twice as high as the current frontline objective response rate to doxorubicin, the single most active agent in soft tissue sarcomas. So our molecular screening program, or MOST, is now open at 11 centres around the country. We have a centre involving the Garvan Institute, where I'm based, and the National Health and Medical Research Council Clinical Trial Centre here in New South Wales. We have a patient advocacy group, Rare Cancers Australia, and a number of other key components of central infrastructure. But we then have a range of cancer centres across the country enrolling what we hope will ultimately be 6,000 patients, of whom we hope to put more than 1,000 onto various clinical trials. To date, we've enrolled about 3,000 patients. They've been referred from more than 100 institutions and from more than 300 clinicians. And I show here the range of different cancer types that have been enrolled in the program. I guess the central point here that I'd like you to pay attention to are the fact that bone and soft tissue sarcomas, because they're such a neglected group of cancers in general, are actually oversampled in our program. Although we don't specifically go out to seek these populations, it's the populations which have greatest levels of unmet need which are enrolled into the program. That's also evidenced by the fact that of the top 10 cancer subtypes, you can see a range of cancers with particularly poor outcomes. For example, brain cancers, pancreatic cancer, lung cancer, cancer of unknown primary are amongst the most common cancer types enrolled in the program. Secondly, you'll notice that we're actually enrolling faster than our projected anticipated recruitment. By this stage, that is to say, the beginning of January 2021, we expected to be at 2,100 odd patients onto the program, but in fact we've been inundated and are now at 3,000 patients. And the original program was projected to enroll a total of 3,000 patients over 5 years. The program goes till 2023, and we project now our ultimate target to be around 6,000 patients. We hope to increase that number over time. So we offer these patients molecular screening, and of the 2,500 or so who've had a molecular tumour board report issued, we've had about 1,500 with a treatment recommendation, and of those patients who've received treatment recommendations, about a total of 60% or so have had an actionable, clinically actionable hit rate, that is, have a recommendation for therapy which is based around some clinical evidence of activity, if not in that cancer type, then in another cancer type. And I show to the right a little bit of how our molecular tumour board works. We have medical oncologists, bioinformaticians, clinical geneticists, as well as our support and cohort team involved in making all these decisions every week. In an average 90-minute session, we would perhaps discuss between 25 and 30 patients to issue recommendations for our patients enrolled onto the MOST program. Following treatment recommendation, we refer on to both our own trials, as shown here, but also to other trials going on around the country which might be relevant. To date, out of the 2,000 odd patients who've been enrolled, 2,500 who've been enrolled and offered a therapy, we've put more than 200 onto our own trials. We started in 2016. You can see the white line is the current suite of trials and our projected suite of trials that will be coming online in 2021. At the moment, we have 2 trials that are dedicated to sarcomas in purple, an interleukin-23 inhibitor called tildrakizumab, based around preclinical research which our own group has developed, and eribulin, a taxane analog which may have activity in epithelioid hemangioendothelioma. But many of these trials are actually accessible to patients with any cancer type, in particular our immunotherapy suite of trials. So far, we've been able to look at outcomes in a subset of these patients who've had more than 12 months of follow-up, and pleasingly, we see similar results to that which I showed previously from Rizal Kurzrock and others around the world, showing that patients who receive a matched therapy, whether on our own program or outside our program, have a significantly improved survival compared to those who do not receive a matched therapy. and the p-values are shown here. It's approximately a doubling of survival, indicating that for the 200-odd patients that we've put onto this program, onto treatments as a consequence of this program, that we've been able to offer something that's meaningful in terms of their options, remembering that this is a population with incurable advanced disease who've run out of other treatment options. In terms of epithelioid hemangioendothelioma, we've got 21 Australian patients enrolled. The program is limited to Australian patients because of the necessity to treat them within our cancer centres, which requires Medicare eligibility. We project we'll have 40 to 50 patients by the end of 2023. The average age is 47 years. The median time of diagnosis, as everybody in this room will be aware, is quite variable. So it's an average of 14 months, but the range is from 1 month to more than 21 years since diagnosis to enrollment on the program, presumably with metastatic disease. 12 of these patients have had prior systemic therapies. 5 have had 2 prior systemic therapies, and 3 have had more than 3 or more prior systemic therapies, which include paclitaxel and, unsurprisingly, doxorubicin. 2 patients have been treated with anti-angiogenic therapies, and then a scattering of other things, which may or may not have been related. In terms of the genomic profiling, we've identified only 2 individuals with pathogenic variants, one of which is a BRCA2 essential splice site variant resulting in a frame shift early in the protein, and probably real. It looks like it's somatic rather than germline in origin, and one a deletion of the gene NRG1. We've enrolled 4 patients to date onto most sub-studies, meaning that about 20% of the expected rate are being enrolled. 2 of those have been enrolled into a doublet immunotherapy trial, and 2 onto the eribulin program, which is specific for patients with angiosarcoma and epithelioid hemangioendothelioma. I don't propose to talk about the results of these patients yet. Suffice to say that one of the patients has had an objective response to doublet immunotherapy, but we're still waiting for accumulation of the results of the other trials. I will say that the eribulin trial is an interesting one, potentially for this group to consider, because it's being done as a split 2-part trial in which we're enrolling 16 patients here in Australia, but there are another 16 patients being enrolled by Dr. Greg Cote at MGH East. The principle behind the study is that taxanes have activity in angiosarcomas and potentially in epithelioid hemangioendothelioma. The novel taxane analog eribulin may have activity, and we're trying to determine whether that's the case in this study. We hope to report out sometime in late 21, early 22. We're developing the program further, and this might indicate where we're thinking that this might head. So I show an additional set of programs here. We have a pancreas sub-program, a lung sub-program, and now a hematology sub-program led by Australian experts in these fields to focus on particularly high priority areas. There are more than 450 patients worth of most sub-studies in development, and 2 of these are relevant to the sarcoma population. I've mentioned the interleukin-23 and the eribulin study, but there will, of course, be other opportunities, larotrectinib, the NTRK inhibitor, and then a variety of other options down the track. We hope to expand our immunotherapy programs in particular. So this is what the lung cancer program, and it indicates the sort of value of a focused effort on particular underserved subgroups. You see the broad range of clinical trials that we have currently available for patients with metastatic lung cancer. These patients go on to existing therapies as shown to the left, and on the right you can see a range of sub-studies which we're developing or have developed, which would offer options to what we call the long tail of actionable targets in these diseases. A similar program has just been approved in pancreatic cancer, which will enroll more than 250 patients with pancreas cancer. I'll just remind you that pancreatic cancer has a 5% 5-year survival. Maybe it's increased to 9% now, but it's still woeful in an area that desperately needs progress. This program offers not only access to our existing range of pan-cancer sub-studies based on the targets you see to the left, but we're developing 2 pancreas-specific trials, which will hopefully fill out the opportunities to ask intelligent and potentially important questions for patients being enrolled onto the program. I think one of the areas where we could develop a further histotype-specific program is in sarcomas. We already have 2 sarcoma-specific trials, but we could develop further trials which have specific activity in this population, including in epithelioid hemangioendothelioma. And you see again the general strategy here is to learn as much as we possibly can about populations which have been typically understudied, but at the same time to offer patients opportunities that they wouldn't otherwise have to receive treatments. I'm going to finish off now by talking about the other very important part of the program, which I haven't had time to go into, which relates to the genetics of the question, why did this happen? It's obvious from both array-based genetic studies over the past 20 years, as well as twin studies, that for many cancer types, and in particular this is true for sarcomas, which occur at a younger age than average, there is a strong genetic component. And of course, we know very little about the genetics behind sarcoma since they're being relatively understudied. You'll notice that amongst the cancer types reported in this overview, that sarcomas are not represented. So through the International Sarcoma Kindred study, we've enrolled 3,000 families across 23 centers in 7 countries. That is continuing to enroll, in part because of rare subpopulations. Epithelioid hemangioendotheliomas comprise 47 patients, or about 1% of the cohort, a little over 1% of the cohort. Of those, we performed whole genome sequencing on 1,600 patients, including 20 patients with the EHE. The average age is 48 years, 12 are female. Interestingly, our pedigree analysis of the kindreds of these individuals who develop EHEs just shows that about 4 would fit conventional criteria for the well-known syndrome Li-Fraumeni, as well as 4 families who have odd patterns which appear to have excess cancer, but where we can't put them into a particular category, and 11 appear to be genuinely sporadic without any significant family history. We haven't found any clear-cut cancer-causing pathogenic variation to date, but we've only got 20 patients. As we get more patients onto the program, and as we undertake whole genome sequencing, we hope to understand more about that. We have developed fairly advanced analytic techniques which allow us not only to catalog existing known variation in known genes, but to discover new genes. And the plan, with the support of the Australian arm of the EHE research community, led by Jonathan Gronek and Jane Biddlecombe, we plan to sequence all 47 patients, undertake such an unbiased analysis to see if we can uncover the genetics behind these diseases. That's really all I wanted to share with you today. I hope you find it interesting, and I'm very happy to take questions offline. I'm sorry I can't be with you in person, but Australia is a long way off in another hemisphere. Good luck with the meeting.